First of all, I'd like to thank you, Ruland, for that nice introduction. And I'd like to thank Lorraine and the organizing committee for inviting me here today. It's a pleasure to be here. And I really enjoyed that introduction talk, Ruland. We come from the same background, I think, in terms of, we'll say no more. So what I was asked to talk about was really the Advantage trial, which is a trial that we've started in pediatrics in the US, and we're going to slowly but surely, hopefully, enroll Canadian centers within this trial. Tom White-Hansen will be leading that effort. So what I'd like to do is to tell you a little bit about that particular trial. That's going to be the focus of what we're going to discuss today. I don't have any disclosures in relationship to this particular talk. So what Lorraine asked me to do was to focus a lot on the challenges and success in starting this trial. And that's sort of a long story. But what I wanted to do today was to sort of focus on a few things. And I think what Ruland talked about just now sort of highlights these issues. So this all started as a result of Tom White-Hansen, Mike Seifert, and I getting together and starting to brainstorm over what it is we needed to do in pediatric transplant in order to sort of build a transformative care initiative. And we formed this group, which we called the Bright C Consortium, which stands for Biomarker Initiative to Advance Transplant Outcomes. And really at the time, and this is now about 5 or 6 years ago, when we met with various folks, everybody wanted to use biomarkers. They wanted to do what Ruland discussed. Let's identify the patients who are going to benefit from our treatments. We're just looking at patients. We're really doing things in an old-fashioned kind of manner. So we put together a few discussion groups, and that was sort of what came out of that. And then I was fortunate to meet a local philanthropist and a foundation who was really interested and had a vision to sort of build a transformative care initiative. And they allowed us all to get together, and we had a little meeting where we discussed a variety of these events, what it was going to look like, what would be a rationale for it, brought in several mechanistic groups. And what I'd like to do today is discuss how it is we built a rationale for what I refer to as a transformative care initiative. So the first thing was certain assumptions. So one of the assumptions that is generally out there is that our current immunosuppressive medications have decreased the incidence of acute rejection, and acute rejection is a problem of the past. But actually, that's not the case. We've decreased the incidence of clinically significant acute rejection, but it turns out that subclinical rejection is still quite prevalent in pediatrics, much more prevalent than it is in adults. And Mike Seifert had just published this study in AJT, which is one of the largest in pediatric groups, where he evaluated biopsy-proven, these were surveillance biopsies performed on pediatric transplants, and as many as a third of them had a subclinical inflammation on their biopsies. And he found that those patients who had this subclinical inflammation did worse over a 5-year period. Now, he used a composite for his endpoint, and the composite was subsequent acute clinical acute rejection, graft failure, and decreasing GFR. So basically, it turns out that a third of our patients are not getting optimal treatment in terms of what we're currently doing. So that then set the stage for saying, well, we need advanced tools. We need to monitor our patients. We need to optimize our therapeutic regimen. And from my perspective, from a scientific, I want to understand the basis for the alloimmune failure. Like, what is it that's different about kids than we know about adults? At the time, the adult groups would say that about 15% of adult kidney transplants would have a subclinical rejection. So it was much more in pediatrics. Some groups felt that there was a need to do a genomic testing for the same reasons as Ruland mentioned, that we're taking a smoothie of people and we're giving them all the same drug. We want to identify what is in that smoothie in order to sort of who would benefit from certain agents. I personally felt a lot about doing full-scale transcriptomics. And Tom, as you'll hear later, was very big on using urine biomarkers in order to optimize our care. The other issue that came up was late acute rejection. So late acute rejection is defined as rejection that occurs after 1 year, so after 12 months, as shown kind of here. And if you look at the era of transplant, late acute rejection has certainly decreased a lot over the past 20 to 30 years. But if you look at the more recent cohorts, that is the golden line here versus the red, which is the more recent one, late acute rejection is on the rise. So why is that? Is that because we're not using our medications correctly? Is that because we're not defining our patients correctly? Or is that because we need some sort of a better monitoring tool? We just let these patients ride, so as to speak. So the other assumption that was made was that chronic graft attrition or chronic rejection is inevitable. All patients are going to get this over time. It's just a matter of time. And if you get it, there's no treatment and there's no way to interrupt the progression of disease. And I personally was following certain studies at the time and thought that is not the case. We can prevent the disease. And one of the drugs that was out there that was quite dramatic in that effect was co-stimulatory blockade. And there were two agents. There's belatacept that has gone through a lot of clinical trials, and there's abatacept, which was actually undergoing a clinical trial centered at Boston Children's Hospital at the time. But these are the results of the benefit trial, which was in adults. And what they found is that donor-specific antibodies basically don't occur in patients who are on belatacept. So here you can see the data that was from the New England Journal paper, that depending on the dose of belatacept, only about 3% of patients had donor-specific antibodies, whereas for adults who received CNIs, around about 11%. We go to pediatrics, it's over 30% of patients at 7 years who have donor-specific antibodies. Some would say it's as many as 40% of patients. So Eileen Chambers, who's at Duke, had started to use belatacept in her pediatric patients, and she followed them for now. She's got some that's gone out over 5 years, and there's literally no alloantibodies in those patients, whereas the control group were all over 20%. So if we're going to build a clinical trial, we want to use a co-stimulatory blockade as part of the trial in order to address this issue. Another issue is the alloimmune response. So the alloimmune response in kids is probably very different than it is in adults, but we adopt everything that we do from adult trials. So we've started to use full transcriptomics to look at patients who are receiving a transplant. So in this, I'm just showing you a sort of a snippet of some data that is currently being submitted at present. But this is transcriptomes, and what you're looking at here is all CD4 T cells that's in the blood of patients divided into subsets. So you've got naive sets, you've got activated, you've got effectors, you've got memory cells, and you've got regs. And these are all the different subsets sort of color-coded. Down here, you can look at, in this particular case, we got 12 transplant recipients that we've just looked at randomly and done full transcriptomics on them and compared them to age-matched healthy controls. These are patients who come into the clinic for other reasons, but are not on immunosuppressants. They were healthy kids coming in for well child checks. And if you look at just T cells, now we look at CD4 T cells all the time. What you can see is that the transplant recipients who are on immunosuppressants have more effector CD4 T cells. So you can sort of see it in the color coding there. And if you look at it here, you can see in this sort of these two different bars that there is more effector T cells in different subsets. So we would expect that. But if you want to use immunosuppressants, at least we know that we're not targeting the particular subset. So what are those doing? We would assume that they're donor reactive. What was also quite interesting is when we look at effector T cells, and this is CD8 T cells, here again, color coding of all the different subsets of T cells, what you can see is that there's a massive expansion of effector CD8 T cells. These are subsets that we don't tend to sort of look at or don't care about so much in our pediatric population. And what's interesting about these subsets is when we sequence their T cell receptor which is sort of coded here in terms of what are these little circles here. It turns out that the expanded CD8 T cells are all of the same clone. So these are very specific donor-reactive T cells that are there in these patients who are so-called stable after transplant. And you can see the populations of cells here. So there's a lot of stuff going on in the immune system that we just know nothing about. And at the time we were doing this, suddenly COVID hit, and we started to look at protective immunity, thinking that there's something really interesting about this. Now, as we know, when the COVID hit, people were dropping all over the place. We were all very worried, and it was all adults with some sort of underlying disease. And the adult kidney transplant programs were quite concerned about it. But what we were finding was that the kids seemed to be doing okay, all right? And actually, when you start to think about it, the kids are exposed to novel viruses throughout their lifespan, whereas the adult groups weren't. So maybe there was something different about these patients. So we started to get blood from all the patients who were getting vaccines. And we saw the same kind of thing that we're seeing in adults. This is looking at antibody to spike proteins. These are the 3 different spike protein responses. And if you see in this case, it's healthy controls. These are healthy kids, and they got their first shot, their second shot, their third shot. And what you're looking at is a very nice response in terms of the response to vaccine. So their protective immunity was good. But in all the transplant recipients, in this particular case, it was some heart transplant recipients, it was one liver transplant recipients, but most of them were kidney transplant recipients. Some developed antibody, but most did not. And we knew that, right? So their protective immunity in terms of responses was not really what we would expect, but they weren't getting disease, right? And what was interesting is that they actually mounted a T cell response. A lot of them mounted a T cell response, but not a B cell response. So here we have the patients, and you can see that we have some we call non-responders. These are patients who had no T cell and no B cell response. And there were some that had a T cell response. In this case, it was 5 out of a total of 12 patients had a T cell response, but not a B cell response. And there were a few that had both a T cell and a B cell response. So there's something funny going on there. And what was also interesting is that there were abnormal B cells in a subgroup of these patients. So the T cells that were present in these patients were interacting with the B cells, but the B cells weren't doing anything. So one of the things that I've often wondered is whether this relates to alloantibody production. So we can get a T cell response and we get alloantibody production. So what is it about our T cells or our B cells? So these were abnormal B cells that we're finding in some of these patients that had the T cell response, but the ones that had nothing had a very different B cell response. This is just to show you, looking at a different way, looking at a thing called CyTOF, which is a protein analysis, but just to show you that our transplant recipients have this very high population, this strange population of B cells. So we're trying to characterize them. Now, as part of a clinical trial, we interact with somebody, Peter Sage, who's a very big T follicular cell person. So there's a subset of T cells called follicular cells, and they interact with B cells. So he would argue that the immunosuppressants that we have are tickling some of these different T cell subsets. And he's particularly interested in a T cell subset, which is known as a regulatory follicular cell. And he's defined a total of 4 different subsets of these cells. And he believes that immunosuppressants target one, both, or many of these subsets. So for instance, in adults, it would be suggested that a CNI would get rid of all these cells. So all the regulatory B cells are gone. That means that only the helper follicular cells are there, and they can tickle the B cells to make, presumably, an antibody. So these cells need to be preserved. So another aspect of a rationale for building a trial is the intragraft microenvironment. And this is something that I'm particularly sort of interested in. And it's an area that sort of has exploded in my world and in the lab. So we know, and it's characteristic, that effector immunity, as I've sort of described, is critical. in terms of the rejection response. So we need to understand this, how it happens, what it is that targets this, and how to inhibit a donor-specific effector alloimmune response. Some people feel that we need to promote immunoregulation at this level, and I don't disagree with them. This is a critical component of rejection or alloimmunity. But what's underappreciated is the role that the graft microenvironment plays. So classically, and from a biomarker perspective, we look at the microenvironment and we say that if the environment is pro-inflammatory, it will adopt this inflammatory response, and so the inflammation occurs locally. However, it's also well known that the graft microenvironment produces immunoevasive factors. And so there's this balance between what is immunoevasive, what I call pro-immunoregulatory, and pro-inflammatory, right? So this is a balance that is constantly being tipped. And it's also becoming clear from the tumor microenvironment that immune evasion can be very potent, okay? And what's also very evident from the tumor microenvironment is it's different in different tissues. So if we start to profile immune evasive mechanisms, we will find that there's going to be organ-specific differences and perhaps families of molecules that can be adopted to address this issue. Now, one of the things that we're personally working on in the lab is the endothelial cell phenotype, because we believe that the endothelial cell phenotype is quite important in this process. And we're starting to look at that in great detail. This is just to give you an example. Folks out there, the current textbooks, would believe that an endothelial cell is an endothelial cell. But over the past 4 to 5 years, we and others have been starting to do single-cell RNA sequencing on endothelial cells from tissues. And there's a whole heterogeneity to these particular cell types. Most of the studies that I've done have identified at least 7 different subtypes of endothelial cells within tissues. We have characterized up to 12 of these subsets, and these papers are starting to come out right now. But this is again a UMAP, and this is a sort of a center of giving you a sense of all of the different endothelial cells that are within a heart. But if you look at classic molecules like ICAM1 or VCAM1, they are only expressed on subsets of these endothelial cells, not on all of them. And what's interesting is that a series and families of immunoregulatory molecules are also expressed on these subsets of endothelial cells. And they are very different in different tissues. This is a violin plot just giving you a sort of a sample of where all these cells are. But if you look at, for instance, VCAM1, you can see that it's dominantly on these subsets, which happen to be more post-capillary venials. And then sort of an immunoregulatory molecule like the galectins is pretty much on all of them. So how these are regulated is going to be really important. So one of the things that we've done is we've gone back and we've adopted a model that was published over 20 years ago by Fadi Lakis's group. And we sort of changed the model a little bit. But we wanted to try and address this in a mouse model to see whether it really is the case. And we're starting to look at humans now, but I want to show you a little bit in the interest of time from the mouse model. So the hypothesis is that the phenotype of the microvascular endothelial cell is a determinant of graft survival. And what we do is we take a fully MHC mismatched heart and we put it into an immunodeficient mouse, and then we give back the immune system at different time points. So if you put the heart into the immunodeficient mouse, it will survive long term, in this particular case, more than 100 days. But if you give the immune system, and in this case, we take alloprime T cells, adoptively transfer them into the mouse, and we adoptively transfer them 2 days after transplant, what happens is you get acute rejection. All the grafts fail. And here you can see the data. The median survival is around about 10 to 11 days. But if you wait And if you wait more than 50 days, in this experiment, we put in the cells on day 70, same cells, you do not get acute rejection. In fact, the grafts just sit there. And in our case, only 2 of them failed over a 30-day period. I think it was like a 28-day period. So the only thing that's different in this experiment is the tissue. Everything else is the same. So there's something about the tissue that can adopt a phenotype that can be immunoevasive. And this is in the transplant type setting. So what we do is we isolate the endothelial cells and we do full transcriptomics. We look at all the genes that are expressed on the cells. And I had speculated that the cells in the day 70 graft would be very quiescent, very much like a donor graft. And the day 2 ones would be a little bit inflammatory from ischemia reperfusion. The day 2 grafts were a little inflamed, don't get me wrong. But the day 70 grafts were highly active. They expressed tons of genes. Like this is the sort of the heat map. I want to get into the list of genes, but there was MHC class 1 and 2 molecules, there were co-stimulatory molecules, there were co-inhibitory molecules. Totally the opposite to what we'd expected. But I do want to show you what we call a gene ontology analysis, looking at the signals and pathways in this sort of 20,000 plus gene set that we were analyzing. And one of the things that's really important here, you're looking at an enrichment score. So if we compare the day 70 to the day 2 graphs, what we find is that there's a lot of interferon gamma responses. Now, interferon gamma is thought to be part of the axis of evil, pro-inflammatory. So there was interferon gamma responsiveness. But what was also quite interesting, if you look in this particular case at the brown and the red line, that there was a marked inhibition of the mTOR pathway. So mTOR pathway was down and gamma signaling was up. And the same basic principle as if you compare the day 70s to a donor graft baseline. So clinically, we thought this is important because if you have a decrease in your mTOR signaling within the tissue, and in this particular case, in the endothelial cells of the tissue, you can develop what I refer to as an immunoevasive graft. So in the context of this sort of model, people have used this sort of seesaw kind of thing, that the activated endothelial cell is bad and the pro-inflammatory, it's down. But actually, I think the model is more like the activated endothelial cells can be pro-inflammatory as a result of certain genes that are expressed. But the activated endothelial cell can be pro-tolerogenic as a result of certain genes that are expressed. So if we're going to build a clinical trial, then what we would suggest is that we'd like to use an mTOR inhibitor as well within the trial. Maybe we'll do that because of all the studies that have been done showing that mTOR inhibition is important in terms of altering an effector and a regulatory cell responsiveness. mTOR inhibitors are thought to enhance peripheral immunoregulation, that is T-regulated effects, inhibiting the proliferation and differentiation of CD4 T cells, enhancing regulatory cell differentiation, and targeting effector T cell activity. A lot of work done on that. Maybe that's a good thing. But perhaps, in addition, you're using an mTOR inhibitor, you would target intragraft endothelial cells or the tissue to become pro-immunoregulatory and allow for the immune system to adapt as it interfaces with the tissue. In fact, in the tumor world, folks have looked at that and they find a very low dose, around about a third of the dose that we would use to target a T cell is biologically active in an endothelial cell. So it's possible, we don't know the dose that we need to use here to target the graft, but I would speculate that we used a very low dose. Like we do know, for instance, if we use the doses that we use for T cells, we have problems with healing. Well, that's angiogenesis, right? But maybe if you use a third of the dose, you'll have an effect on the endothelial cells or in the tissue. This is something that I think needs to be discussed or certainly needs to be evaluated. So based on this meeting and these sort of rationales that all came out of this meeting, we would say that the overall goals, if we were going to build some sort of a clinical trial initiative that we wanted to be transformative, would be for pediatrics to prevent alloantibody production, to promote immunoregulation, and to target the subclinical rejection response, perhaps, as I've mentioned, through targeting the graft itself. That these events would have selective targeting of the alloimmune response that should be pediatric specific, because there are different issues from a pediatric perspective. And then we would define the mechanism of action of our drugs using omic approaches, multiple omic approaches. And we can look at the efficacy of what we're doing using biomarkers, particularly the urine biomarkers that Tom's going to talk to you about. So that's what we did. So the hypothesis that we generated is that the early introduction, when I say early, literally on day 1, of belatacept and sirolimus as an immunosuppressive regimen will be safe and efficacious to prevent the donor-specific antibody production, augment immunoregulation, and enhance long-term function. The trial that we built and that we started right now is as follows. So in order to do belatacept, we need to have EBV seropositive patients. It'll be deceased and living donors, age 6 to 20. I wanted to go down smaller, but with the EBV issue, I think the younger kids are going to be difficult. And there'll be a control arm, which receives standard of care, which will be TAC and MMF. And then the intervention arm will receive the sirolimus. Because of the healing issue, folks did not want to start the sirolimus on day 0, like with the Bella. So we're going to use a TAC bridge for 2 weeks. And then we want everybody on the sirolimus by the end of the month. The goal here is to get these drugs on board very early, before the immune response is established. So you're targeting it right de novo. And then what we'll do is we'll profile the alloimmune response, we'll profile the graft, we'll perform biomarker assays, as well as additional mechanistic assays, and see what we're doing. We're not going to wait 1 to 2 years and sort of say, yep, we're patting each other on the back, we're doing a great job. We want to know exactly what we're doing. And the primary endpoint is DSA at 1 year. And we're expecting that these patients who are on belatacept should have less than 5% of them should have DSA. We actually think it's going to be closer to 0 based on the pilot studies that we've done. And then we look at GFR at 2 years to see whether or not we're making any difference from that perspective. This is what it looks like from a practical perspective. And this is where I'm going to tell you a little bit about the challenges in building this consortium. But this is a collaboration that we've built with the NIH. And we have a data safety monitoring board, which is also within the NIH. There's a clinical group, and this is chaired by Eileen Chambers, who has a lot of experience using belatacept and did the early trials with this. Nancy Rodig is also a chair, and Sandy and Mike Seifert are co-chairs. From a biomarker perspective, Tom and I will look at that. We're particularly interested in building urine biomarker assays. And then Jay Fishman, who's an infectious disease expert at the MGH, and I will help work on the mechanistic side. Jay is very interested in the infectious disease issues, as I discussed earlier. And right now, we have about 20 centers, but we're probably going to need more. And we have 10 core laboratories performing all of the studies that I just described to you. Pete Sage, that I mentioned, is also going to do a trial of influenza vaccine. So all the patients will be receiving influenza vaccine. So what we would expect is that there's going to be differences in terms of their follicular cells, whether they respond to vaccine or not. And that'll help us in terms of analyzing the protective immunity. But the overall approach is an integrated approach between what I refer to as clinical trialists and mechanistic groups. And just to shout out, we're finally at a point where we're going to work with Health Canada and try to get Canadian centers involved. And Tom is going to lead this. So at a certain point, if you're interested, talk to Tom. We want you in the study. I'm going to finish with just mentioning a little bit about the urine biomarkers, because I know Tom is going to talk a lot about this later. But as I mentioned to you, one of the things that I think we're very deficient in the pediatric world is we just monitor creatinine. We're literally still on flip phones. You know, this is what I tell people is if you walked into your doctor and he was on a flip phone talking to somebody, you'd say, geez, you're a little old fashioned. That's kind of what we're doing here. It's kind of crazy. And one of the things that we've got access to are these automated assays for looking at biomarkers. And so both Tom, Mike, and I have been using this system. This is an automated system, which was originally made by a guy called Marty Putman, who ran this protein simple assay. I knew Marty as he built this. And we started to measure 4 biomarkers, CXCL9, CXCL10, because they've been studied for many years. They've been shown to be increased in association with rejection. And we looked at 2 others, CCL2 and VEGF-A, because they may be important in more of a chronic inflammatory response. This is an automated system. You build this cassette, you stick 25 microliters of urine in it, stick it in a machine, an hour later, you get a result. Totally automated. And what's interesting is we've studied this in over 500 patients at this point in time. And what we get is, and this is just to show you a very close correlation in the urine between CXCL9 and CXCL10. So this is showing you the increase in both of them. And if you look at the red, which is rejection, you can see that most of the patients with acute rejection are up in this quadrant. So both of them go up in rejection. For borderlines, in this particular case, the borderline was pre-2019 Banff borderlines. It was all over the place. And the normals tended to be different than the acutes. But this is just to give you a sense of the way this assay can work. We also did analysis just looking at standard ROCs for rejection, and the ROCs were pretty good at around about 0.83, and if we use it with creatinine, about 0.85. So this assay seems to work. What I want to show you is if we use it in the clinic and we start to monitor these patients, and we've done this at Boston Children's over the course of a year. So we've taken various patients and we've been collecting their urine serially and then measuring it. So these are all patients with normal serum creatinine. And we're not doing surveillance biopsies, although this is stimulating them to do surveillance biopsies at Boston Children's. All these patients, normal serum creatinine, normal serum creatinine. These are the patterns that you get. These are cutoff levels for CXCL9 and CXCL10. So this patient always had sort of lowish levels. And if we look at a score 2, which is an analysis based on our algorithm for the 500 patients, you can see that their risk is very low here. And if we look at score 4, all 4, this particular patient, the risk is quite low. But this is another pattern that we get in another patient. This patient tends to have very high CXCL9 and high-ish bouncing around CXCL10s. And if we look at the score for these, it's going up. So we would suggest that this is an at-risk patient based on the urine biomarkers. I think Tom would agree with that, and I'm waiting to hear his talk, but we'll talk about that later. So that's what we're doing in the trial. It's just started. It's going to go on for at least another 3 years. We think we're going to need 2 years to enroll all the patients. We're going to need a lot of centers, and we're building. So what were the challenges and why is it taking so long? I think that Roland sort of pointed to every single one of these issues. So when we started this trial, I met with everybody. I said, we want this up and running within a year. And they looked at me like I was crazy. And I said, no, we're going to do it, right? But actually, they were right. So in the first year, spent a lot of time finalizing the clinical protocol. We have to develop a new drug application, which goes to the FDA. We submitted that to the FDA, and we worked with industry to develop sort of means of getting the drug to all these different centers. And then you have to build standard of practice for sample processing and shipping them. Then the FDA come back, and they're very careful about pediatrics. Oh my gosh, in pediatrics, kidneys are precious. You can't do this, right? It's just crazy, right? So we had to prepare a response to them. We had to come back to them And cut a long story short, it took about another 6 months into year 2, and we got the approval. So once you get the approval from the FDA, you then go to the IRB. You have to get your IRB approval. At that particular point, we start to build databases, randomization issues, and management of, various events that need to be reported to the Data Safety Monitoring Board. So in year 3, then, the IRB's approval, so then we start the process. And that's kind of where we are now, sort of building this. We completed all the regulatory stuff, and we have started to enroll patients. So it took 3 years to develop the trial, which is just crazy. But it's all because of these regulatory responses. So in developing pediatric trial, the successes are building strong rationale for the trial through transformative care initiatives. I think we want to include mechanistic studies in anything we do. Successes are also in terms of building these consortia. And I agree entirely what Luann said. We can't do this alone. Bringing everybody together is the only way to build this, and getting lots of patients enrolled. The challenges have been all these regulations around the pediatric patients with the FDA, and it moves a lot slower than we would have thought. So I'm going to finish there. Just want to acknowledge the folks that sort of have helped with this. This is a collaboration with the NIH. Jonah Odom is the clinical section chief at NIAID for transplantation. And he's been amazing in terms of helping to sort of build this process. And we have a program manager and we have a regulatory manager. And Julia will be working with Tom in terms of sort of pulling this whole thing together. She's been great in sort of helping us to sort of work with FDA and now with Health Canada. And the clinical leadership, as I've mentioned, which is Eileen and Nancy. And then the mechanistic leadership, Tom, Mike, and I have sort of spent a lot of time working on this, and all the other folks. I just want to mention the informatics group. We're very fortunate to have some really good folks here. Alvin Coe has just been great and totally data. If anybody wants to do anything with data, we can put Alvin on it to help you. And then, of course, the funding. So I've been fortunate in terms of initiating all of this to have these foundations available. Like obviously you have some foundations available as well, but it's only through the vision of these foundations that we can jumpstart this sort of stuff. Thank you very much.
Thanks so much. Thank you so much. I think we're open for questions. And I don't know, are we going to get questions from offsite also? People are virtual. They'll come, or they'll be here. Okay, great. Perhaps I'll start off, if there are any questions here, but I'll just start off the discussion. I was wondering, I actually had lots of questions because I wrote notes, but I wanted to ask, when you're thinking about, you were talking focus primarily on kidney transplants. When you're thinking about endothelial cells as potential immune mediators in some way, do you think that there would be rationale then to think about the heart transplant patients a little bit differently than the kidney transplant patients? And so what do you think about that?
Actually, we were just talking about that. I think I'd love to talk with Laurie about this a little bit as well. So it's very interesting, but when you do single cell RNA sequencing, the endothelial cells of each organ take on a phenotype related to the organ. So endothelial cells within the heart can express cardiac myosin. Endothelial cells within the kidney can express transporters. Okay, endothelial cells within the lung express a whole other variety of molecules. And this is out there. This is not published by us. This is published by others. So if you think about the immune microenvironment that you're building, it may be that there's a whole series of events that are happening. And from what I understand, you're starting to look at the microenvironment in terms of changing from an ABO perspective. It may very well be there's a lot of other things changing in the heart as well. It'd be fun to actually look at that a little bit more. So absolutely. And I think that the concept that we're throwing out here from the point of view of targeting the tissue could actually be organ specific. I don't know about heart and lung because we've only done it in kidneys, but there is data that we've looked at in kidney patients who are, quote, tolerant. And one of the key features when you do full transcriptomics on their tissue is that the mTOR pathway is really low. So I would assume, because of the mouse data, that it's similar to the heart in the heart, but I don't know. But yeah, very good question. I think we really need to look at that.
Simon.
Congratulations, great planned trial. I had one question for you, because you're switching out two things, right? You're moving away from the calcineurin inhibitors and moving from the mycophenolate to self-effective mTOR inhibitors. Why not have a third group that mixes CNIs with mTOR inhibitors so that you know which makes the inhibitors?
So I'm just going to repeat the question for the people online, just to say, Dr. Urschel was asking, why not have a third group in the clinical trial that included MMF and tacrolimus as another comparison group? Oh, so sirolimus and as another comparison group.
Actually, we wanted to do that, but the NIH are very focused on getting a very simple trial. Like, it's hard enough to get the trial going as it is. So you just need to have two groups. I think that's particularly important for the hearts, because a lot of folks do want to do that. Kevin Daly, you may know Kevin Daly. So he's built his trial using everolimus, but I think they use a little tack there as well. That's the trial. So yeah, we can start to think about that a little bit too.
Next question, Lori.
Terrific talk, as always. And both you and Lynn have nicely pointed out all the difficulties and challenges of doing these sorts of studies in the pediatric population. But I have a question for you about, you're covering a range age 6 to 20 that covers a lot of physiologic change from a developmental perspective that adults don't face either. So what's the best way to analyze the impact of, for example, sex and adolescence in a child who's between 6 and say 17? How do you propose to look at those things and the impact on that?
I'm just going to repeat the question for the people online. Dr. West was just asking, the clinical trial includes participants ages 6 to 20. And the question is, what's the difference of sex as well as aging and adolescence on immune function, and why that criteria given the impact on immune function?
So the answer is yes, but I would add in race. So yes, so that is something that we need to do. At this particular point in time, it's going to be a power analysis. Like, we just got to get the patients enrolled. Like, we just barely have enough power with 200 patients. And that's one of the big problems also with kids. Like, I think there's two other initiatives that we have going now. There's one called the Genomic Information Commons that is headed up by Ken Mandel, and we're involved in that. This is a process where we're sequencing, doing full exome sequencing on all patients who come to clinics at 4 different centers in the USA. And so we will have exomes on every one of our transplant patients. Right now, it's over 600 patients that we have. There's 1.4 million kids enrolled in this study. And so once we start getting these types of data back, then we'll start to be able to look at that. The problem is going to be getting access to the electronic health record in a de-identified manner to be able to sort of see how it affects outcome and other analyses. But this would be a discussion that you, Rulan, and I should have with Ken about what we can do with the data we're going to get from this particular study.
It's interesting. I was thinking the same question. I was wondering, on the 500 patients that you have with the CXCL9 and 10, whether you and Tom can actually look at sex, age, and then think if there's, an implication for also donor-specific antibody. Because it was interesting, you said 30 to 40% of children get it, but I was wondering, who are those 30 to 40%? And who are the 30 to 40%? And who are those who don't get it, right? And maybe truly tolerize. And so it's interesting because you've taken a blanket approach in the trial, which makes sense. But I wonder if if the success may be hindered, not just based on power, but because that group who actually formed the antibody may not be selected enough in that 200. And that's just one of the things that I worry about. All clinical trials are important, but I just worry about, I don't know if we know well enough. And I don't know if you want to answer, but I don't know if we know well
I
enough the
would just
criteria.
add race in there, like
And
that
then
is,
race also,
race is massive.
effect also.
Like Alvin, who's doing a lot of this stuff, he won't let us touch anything without looking at race. It changes everything.
Really?
Changes it. Tom knows that from some of the studies. It changes everything. Just bear that in mind as well.
Okay, other questions? Yes, thank you.
Thank you, that's great talk. My question is related around, we talked a lot about development of antibody, creating HLA antibody for the most part, although maybe there's other things specific to. The history medical community is working really hard to try to standardize how we're doing things, how we're reporting it. We still have some work to do there. With so many centers participating, curious how you're going to try to standardize the HLA data that you get from so many different sites, or kind of a centralized site, or how do you want to tackle that heterogeneity that might affect some of these outcomes?
Yeah.
So I'm just going to repeat the question quickly, just to say the question is how to plan that standardization of HLA antibodies throughout the clinical trial.
Yep. So the way we've dealt with that is with centralized course. So, you know, Elaine Reed is going to be the core for all of the anti-HLA measurements. But there are, as you know, well differences across centers, but also for pathology with a central core for that as well.
We've got time for one more question.
Well, hi, my name is Michael Florian, so first, thank you for all you and your teams are work to get this trial off the ground. Immense amount of honor out there. I may have missed this in your protocol. How would you address different programs and approaches to transplanting across USA? So if, you know, some centers may more readily transplant the patient present their history more active in the same, will that change the approach to immunosuppression post-transplant? And how do you anticipate it would have an impact on the effectiveness of various immunosuppressive regimens?
So the question was how to handle the heterogeneity, I guess, of donor-specific antibody at time of enrollment in the clinical trial and impact.
So for the enrollment criteria for this, the patients need to be non-sensitized. So there isn't an issue here. Although I will say that there are folks that are using Bella, certainly in the adult group, for sensitized individuals because it does decrease the titer over time, supposedly, but we haven't tested that in kids.
Thanks so much, David. I think this is so exciting and great to see such a comprehensive trial. For all those Canadian sites that are hopefully virtually or present in the room, please make sure you join up and contact Tom so that we could all participate in the trial. So I think this ends our first session, and we're going to go on to a break. Okay, coffee. All right, thank you very much.